Vanakkam. Welcome to this course on classics in neuroscience. In this video and in the following videos, we will be looking at physiology, specifically physiology of synaptic potentials and receptor potentials. Previously, in the previous week, we saw the physiology of the action potential, how nerve impulse is getting transmitted. So, in this week we start our discussion on how synapse works and how receptors perceive or receptors receive this information. Shown here again is these neurotransmitters in vesicles that are getting released and that are being sensed by the receptors. In this class, we will look at the synapse as a functional concept, and we will discuss the first electrophysiological evidence for synaptic transmission and how the advent of intracellular recordings made a huge impact in this field. Remember, in the previous weeks we suggested that the original neuron doctrine was proposed by Ramon Cajal, and he suggested that there was discontinuity between neurons that communicated. How the neurons communicated is something that Ramon Cajal did not discuss in great detail. This was in response to Golgi's reticularism, where there were not discontinuities but rather continuation of neurons. We also saw in the previous week that it was Sherrington who coined the term synapse with the help of a colleague. You can revisit the previous videos exactly who coined the term. But it was Sherrington who was asked to author the neurophysiology chapter and who suggested the name synapse for the connections that happen between neurons or excitable cells. A question that arose was whether synapse is an anatomical construct or is it a physiological construct. Anatomical means morphological or structural, or physiological means functional. This kind of questions happen not just in synapse. For example, in biomechanics there is this question about whether muscle is an anatomical construct or is it a physiological construct. That is it a structural or morphological construct or is it a functional construct? These kind of questions are spread throughout biology, and it is not uncommon to see this in other domains of biology as well. So, the question that haunted scientists in the early 1900s was whether the synapse, is it an anatomical construct or is it a physiological construct. It is likely both, but it is possible for one to be anatomically something and functionally something else. So, physiologically something else, this is a possibility. So, the question of is this an anatomical or physiological construct was the question that went through Sherrington's mind. But by the time he authored the 1906 work on the integrative action of the nervous system, integrative action of the nervous system, I think it was 1906, he was somewhat convinced that it is a physiological concept, which is why he outlines the functions of the surface of separation. He outlines the functions of the surface of separation, which I am going to read out from the text. This surface of separation, he says, such a surface might restrain diffusion, bank up osmotic pressure, restrict the movement of ions, accumulate electric charges, support a double electric layer, alter in shape and surface tension with changes in difference of potential, or intervene as a membrane between dilute solutions of electrolytes of different concentration or colloidal suspensions with different sign of change. So, this shows that Sherrington, who was working at the systems level, was indeed aware of membrane properties of the excitable cells, that he was speaking about electrolyte concentrations, he was speaking about potential differences, but he did not, for example, speak about receptive substances that was suggested by pharmacologists, or nor did he speak about the electrical induction mechanism of how the communication might happen. Remember, in one of the previous weeks we discussed how communication between neurons might happen without contact, like electricity is induced in an inductor, we said that. So, Ramonika Hall suggested that it might be like electrical induction mechanism, but Sherrington, just 15-16 years from the time of Ramonika Hall's neuron doctrine, did not actually speak about these, but he did mention about electrolytes of different concentrations. He did mention about potential difference. That means he was keenly aware of the Bernstein-Overton theory of the membrane potential. Remember the work of Bernstein on plant membrane potentials. We mentioned this in one of the previous weeks. Approximately this is where the matters stood for about 4 decades until about the late 1930s. From about early 1900s to late 1930s, about 35-40 years, this is where the topic got stalled, because this is not a topic that can be studied by anatomists, pharmacologists who stimulate an entire organ and then measure the chemical output. Remember, in the good old days, the stimulation was done at the organ level, not at the cellular level. Much later, only in the 1930s and 1940s, is when individual neuronal stimulation became possible. So, stimulating the brain as a whole was considered weird or considered too complex, and the brain itself is considered seemingly impregnable, impenetrable. So, it is not clear what this means. So, here we show again how communication of neurotransmitters happens through vesicles and exocytosis. So, what is zoomed out here is this area where neurons communicate with each other. So, axon collaterals communicate with dendrites of the other neuron, and of course, the dendritic trees of different neurons are different and are some of them may be very complex or complicated. For example, Google about the Purkinje cell or Purkinje neuron, Purkinje cell in the cerebellum. The dendritic tree is very complicated. I mean, what's shown here is a relatively simple dendritic tree in comparison to, for example, the dendrites of a Purkinje cell. So, the brain may be considered too complex and seemingly very difficult to understand and penetrate. The question of how communication happens between neurons or between excitable cells was first attacked by using the peripheral system until the late 1930s for the advent of microelectrodes. With the introduction of microelectrodes to record, recordings were made from peripheral nerve preparations, mainly the neuromuscular junction and sympathetic nerve ganglion. Several groups worked on this topic. Gopfert and Schaefer in Germany, Feng in China and Eccles and his colleagues. Of course, initially Eccles was in Australia, later he went to New Zealand, but initially Australia. I think that was Eccles and O'Connor. The paper was Eccles and O. Stimulation of the motor neuron initially resulted in a minor or a small end plate potential, right, which later led to an action potential. This was the first example of a recording of a synaptic response and first direct recording of the physiological effect of a synapse. So, you are stimulating the neuron, you are stimulating the neuron here and you are recording from here. What is shown here is sampling of the same neuron, but recording is happening on the post synaptic muscle cell. That is what is being measured. So, initial response is an end plate potential, which further leads to the development of an action potential in the muscle fiber. The details of how a neuromuscular junction, at the neuromuscular junction, development of an end plate potential leads to an action potential to muscle contraction, I will share a different video from the neuroscience of human movements course, that is on excitation contraction coupling. Excitation contraction coupling and sliding filament theory. There are two videos, I think, from my other course. My students will share that with you in forum and in the description. This evidence of the end plate potential was convincing. Remember, Eccles was a strong proponent of the electrical communication theory. So, even he had to agree that no, I mean, communication between excitable cells, at least in the peripheral system, appears to happen by chemical transmission, and because of this demonstration of this end plate potential. How exactly is this end plate potential getting developed? Because of the release of acetylcholine from the presynaptic terminal. Remember from our previous videos, this was demonstrated quite some time ago actually, the presence of acetylcholine, and then there was the work of Kotia and George Coyle demonstrating the presence of acetylcholine esterase at the neuromuscular junction, and so many other demonstrations, so many other evidence to suggest the release of acetylcholine from the presynaptic terminal. But a question remained, okay, this is how the peripheral system works. Is it that the central system also work like this? So, the question of how the central system works remained an open question. So, Eccles continued to hold on to his view that perhaps communication between neurons in the central system happens via electrical means and not via the chemical transmission. So, that had to wait, that communication between central neurons or within the brain, for example, also happens via chemicals, is something that needed further evidence. So, that question continued to remain open. Also remember our previous discussion about soup versus sparks. So, it was possible to record with the advent of microelectrodes and the development of amplifier technology to record from single cells. So, this technique came to be of course called as intracellular recordings, but recording from within the brain cells is challenging, because unlike the peripheral neurons, neurons within the brain are very small, the diameter is small, the length of the axons are also small. Many of these neurons are actually interneurons. So, most of these neurons are really small, because interneurons just take information from one point and either excite or inhibit another part of the brain, mostly within the same subcircuit of the brain. So, this is how interneurons function. Most neurons within the brain are interneurons and they are very small both in length and also in diameter. So, there are real challenges in recording from central neurons, in this case essentially from neurons of the brain. Of course, development of intracellular recording equipment, also the development of the cathode ray oscilloscope, the CROs. Remember, we discussed this in the previous chapters or the previous videos, where we saw how the Americans or scientists working in America developed the cathode ray oscilloscope, and how Hodgkin was amazed in the 1930s and the 1940s primarily, and how Hodgkin's work took a leap when he implemented these principles in his own experiments. We saw these things in the previous set of videos. Remember, the earlier intracellular recordings were made with micropipettes containing electrolyte solutions, but this required the micropipettes to be made using some standard techniques, to be of some standard dimensions. So, the concept of microforge had to be developed. Remember, these are things that we have discussed in the previous weeks. With the advent of intracellular recordings, it was possible that we could measure membrane mechanisms underlying action potential within the cell, is it not? So, it was possible to make these measurements. The first demonstration of this kind of work was, of course, by Ling and Gerard, who showed recording of membrane potential of the muscle, and it was Hodgkin and Nastuk who made the first recordings of the muscle action potential. So, the first model synapse that was under discussion, that was under consideration, was the neuromuscular junction. The advent of this technology and this method, also the advent of the voltage clamp method together, essentially opened a whole new world of intracellular recording and cell neurophysiology, or neurophysiology performed at the level of individual cells or individual neurons. So, with this we come to the end of this video. In this video, we saw whether the synapse is a anatomical or physiological concept, and we see that Sherrington actually defined this as a functional concept, and we saw the first electrophysiological evidence for synaptic transmission performed by many people simultaneously. Even Eccles had to agree that in the neuromuscular junction, at least in the peripheral system, communication happens chemically. But the question of what happens in the central neurons still remained open, and how the whole new world of intracellular recordings, especially in central neurons, became possible in the 1950s. With this, we come to the end of this discussion. Thank you very much for your attention.